Hi, my name is Zimeng Ye. I'm the first author of this paper entitled Cerebrospinal Fluid Liquid Biopsy for Detection of Somatic Mosaicism in Brain. Somatic mutations as a mutation happen after fertilization and are present only in a subset of cells of an individual. If they happen early, they can affect multiple tissues, and if they happen later in the early development, they can only affect only one tissue, such as the brain. Somatic mutations in the brain have been implicated causing epilepsy and brain malformations. But currently, the only established method to study the somatic mosaicism in the brain is by sequencing the brain tissue acquired from neurosurgery or autopsy, which is unavailable for the vast majority of patients. In our study, we are trying to establish a novel strategy by using CSF liquid biopsy to study the somatic mosaicism in the brain. And in our study, we aim to investigate if CSF cell-free DNA can be used to detect brain mosaicism. To achieve this, we need to answer three major research questions. First, is there enough cell-free DNA in CSF to make a liquid biopsy viable? Second, can we use CSF cell-free DNA to detect somatic mutations in the brain? At last, can we show that CSF cell-free DNA is largely brain-derived? To answer the first question, if there's enough cell-free DNA in CSF, we recruited 28 patients with epilepsy and 28 controls. CSF were collected by dural puncture in patients and by lumbar puncture in controls. We then use droplet digital PCR, which is an absolute quantitation method to study the cell-free DNA level. And we found the median level of cell-free DNA concentration in patients is 502 copies per ml CSF and is 61 copies per ml CSF in controls, which shows there is enough cell-free DNA to make the downstream analysis possible. To answer the second question, if cell-free DNA in CSF can be used for molecular diagnosis, we performed genetic testing in 3 patients with focal epilepsy. Each patient has different subtypes of brain malformations and has specific genetic variants. We collected the paired brain, blood, CSF DNA samples from each patient and designed 3 individual ddPCR assays to target at specific mutations. For the first patient with subcortical band heterotopia, her LIS1 variant was previously reported to be 13% in blood and 5% in the brain. And we tested the CSF using ddPCR, and the blue droplets are the ones positive for mutant allele, and the green droplets are positive for wild-type allele. Variant allele frequency was calculated using mutant droplets divide total droplets. And in this patient's CSF, we found this LIS1 variant is 9.4% variant allele frequency. In the second patient with focal cortical dysplasia type 2B, this THC1 variant was 2.8% variant allele frequency in brain and not detected in blood. And in the CSF sample, this variant was detected to be at 7.8% variant allele frequency. In the last patient with ganglioma, before we tested this patient, we did not know what variant does he have. We simply tested this BRAF V600E variant because it is the most recurrent mutation in patients with ganglioma. This variant was initially detected in CSF cell-free DNA at 3.2% and subsequently validated in brain at 20.8% and absent in blood. To answer the third question, if the cell-free DNA in CSF is mainly derived from the brain, we performed the cell of origin analysis. We conducted whole genome bisulfite sequencing on the cell-free DNA and analyzed for the brain-specific methylation patterns. We found CSF cell-free DNA is enriched for the neuronal methylation patterns, which shows that the CSF cell-free DNA is mainly derived from the brain tissues. In conclusion, Our study provides a proof of concept that the CSF liquid biopsy is a viable surrogate for brain tissue to establish molecular diagnosis of brain somatic mosaicism. And in the future studies, we will try to establish genome-wide analysis of the CSF cell-free DNA. And thank you so much.